Okay, I guess we can get started, right, Zoom?
Oh, yes, please, Chung.
Okay, yeah, okay, welcome everybody. And today we're quite pleased to have Professor Tzung Hsiai from UCLA to come here to give us a talk about some of his work. And we're all very excited about that because Professor Hsiai has done a lot of very good work and has been very accomplished. Professor Hsiai received his undergraduate training at Columbia University and medical training in the University of Chicago. And then he did his medical internship, residency, and fellowship at UCLA, where he actually developed some microsensors and studied mechanotransduction of vascular development and repair. He is very multidisciplinary, and he works and collaborates very widely, and he's the key to forming a lot of teams that does that, including one that collaborates across Caltech, Cornell, Emory, Georgia Tech, Mayo Clinic, Stanford, USC, and UCSD, and hopefully eventually includes Imperial College too. He has been taking a lot of professional positions, including the chair of the NIH 3D Printing Studies Section, AHA Scientific Planning Committee. He's been elected to the American Society for Clinical Investigation, and he's a fellow of the American College of Cardiology, fellow of the American Heart Association, and of the AIMBE, which is the American Institute of Medical and Biological Engineering, and also a fellow of the Biomedical Engineering Society, just to name a few out of his many accomplishments. And also an award by AHA, the John Simpson Outstanding Research Achievement Award. And this is why we're all very excited, to hear from him, and very honored that he's here today. So without further delay, Dr. Hsiai, please proceed.
Well, thank you, Dr. Chung, for your very kind invitation, and I feel very honored to be back to Imperial College. And 2 years ago, and Dr. Peter Weinberg organized a very successful meeting on biomechanics and cardiovascular disease in London Imperial College. And I had the first taste of this incredible institution and faculty and students. So once again, thank you so much. Hopefully, this will be an impetus to cross the Atlantic Ocean and forge collaboration with your institution. Congratulations to Dr. Chubb and Julian Vermont for your science publication. We were very excited about your innovative approach to mechanotransduction, leading to the understanding of piezoreceptor calcium influx. I think that our students in the US have been very much interested in pursuing in this direction. And lady, once again, congratulations on your post-biology publication focusing on endo-MT or SNAIL1 and TWIST1, and further providing mechanism to our investigation into valvular formations during endo-MT or endothelial mesenchymal transitions. Having said that, well, I think we hope to see the light at the end of COVID-19 pandemics. But here I'd like to share with you the tool that we've been developing and our humble journey using laser light sheet and recently light field in combinations. Well, the desire to pursue this new optical tool is really inspired by Dr. Julia Vermont's work in embryology and cardiomorphogenesis. And shown here is the embryonic stages starting from fertilized eggs to the early post-implantation, followed by different stages of organogenesis. So we asked the question, what kind of animal model will be more suitable to study cardiomorphogenesis? And shown here are different animal models well known to the community, from fruit flies to zebrafish to avian, as well as the most common model, mouse. and cell culture and mitral as well as organoids. However, in zebrafish, the strength is really to recapitulate cardiomorphogenesis in terms of genetic manipulations. So today I'd like to show you how genetic manipulation to understand mechanobiology. And I think Dr. Vermont is really the leader working in collaboration with Dr. Yap. Well, one or two people is this embryonic model containing of two chambers and atrium and ventricle. However, despite these two chambers, adult zebrafish develop a morphologic cardiac transduction analogous to human EKG. Shown here is the human ECG. And this is the one we recorded in the lab from zebrafish, providing a basis to use this model to study electrical conduction and mechanical contractions. Well, the other advantage is this short developmental stages, starting at 20 hours after fertilization. By 120-hour post-fertilization, one will have a morphologic heart. So one can study, and similar to what Dr. Yap and Dr. Vermont have been engaging in this area, and to assess fluid dynamics during the embryonic tubular formation, followed by cardiac looping and valvular formation. So these stages could be captured. This was the old data taken more than 10 years ago, but it's still good to demonstrate the short developmental stages. So now by 90 hours, one have two chamber with the unidirectional flow by this morphologic AV valve, the valve between atrium and ventricle. Well, we started this humble journey inspired by Ken Paus and his colleague, Alan Niam, because of these two chamber hearts, atrium and ventricle, packed with trabecular muscular ridges and the electrical conduction similar to humans. Likewise, people know about the capacity for zebrafish to regenerate after ventricular resection. So shown here is the entire ventricle, and after amputation to the apical area, morphologic ventricular structure ensue after 60 days post-amputations. So we're very interested in understanding morphologic sequences, and furthermore, studying from the cardiac crescent and to heart tube leading to cardiac looping, and finally remodeling. So I'd like to focus on this area during cardiac looping, and we have two events. One is trabeculations, and the other one is endocardial cushion formation leading to valve formation. So I will first focus on trabeculations. So the talk is divided into two sections, and I have a lot of slides, but I'll be happy to stop for questions or finish early so we can have discussions, and I'd be happy to share other information in the near future. So first of all, we'd like to discuss about 4D light sheet fluorescent microscopy to investigate the initiation of trabeculation and alpha tract morphogenesis. If time allows, we will quickly discuss how we can integrate light sheet with our new light field to study segmental myocardial function and intracardial flow dynamics. Well, introduction about microscopy or optics. This cartoon illustrates the limitation of light penetration into deep tissue. So shown here is a laser light by undergoing dispersion after 1 millimeters. And so you're into this idea of issue with mean free path when light when photon travel into the tissue. This transport mean free path is really limited by tissue property. And this region, the ballistic region, is where microscopy is used for our daily research. However, deeper penetration requires tomography or in the diffusive region. The question is, how can we overcome this quasi-diffusive region for microscopy using light sheet imaging and the combination of optical clearance, and so light can penetrate deeper. This cartoon illustrates the current state of imaging system. As shown here is the light source, and this is the naked eye. So if you have a cardiac tissue right here, notice the mean free path has been disturbed because the tissue property, and so that one will not be able to visualize the light source. Well, to address this is by surgical operation, right? So we have to dissect the heart, and so we can expose the heart to allow for the mean-free pathways. Well, with the use of light sheet microscopy, supported by the chemical clearance to render the tissue transparent, and then this similar idea could be materialized without resecting the hearts. Well, this is the new emerging tool on the horizon for the acoustics and without resection, so one can perform live tissue imaging and dynamic blood flow without optical clearance either. But I think the focus today is to address optical clearance using light sheet microscopy. Well, this cartoon illustrates the fundamental apparatus to demonstrate light sheet microscopy principle. Well, shown here is the microscope, in the cartoon showing the light source starting from illumination in terms of a light sheet, a laser blade that could be cutting across the tissue in 2D. And then perpendicular to this light source is the detection lens shown in the blue, in a green color. So this is the setup where light source will be illuminating the sample, and then the detection will form a perpendicular pathway. And so right here is the sample detected by CMOS camera, allowing for high frame rates per second. And then other optical components allow us to formalize or shape the laser light into a laser sheet. And one can have different laser sources and in terms of different wavelengths for green fluorescent, disarray, or other colorations. So this is the optical bench one can set up in our lab or in your lab, and where light source will be channeled through multiple optical components to generate a laser light sheet to illuminate the sample. And perpendicular to this light source is the detection lens detected by very fast CMOS camera. So I think the fundamental principle is this perpendicular pathway, and so that one can perform intact sample without dissections. Unlike confocal, we have to perform stitching. This is the case where sample can be illuminated by light sheet without stitching. Okay, so this is the example of this optical pathway. And so this cartoon illustrates the laser light sheets crossing the embryo rapidly to generate multiple images. So these stacked images can then be reconstructed into a 3D moving cardiac dynamics. So that's the concept where illumination and detection to generate different stacks of 2D images, and by super resolution or different methods of reconstruction the images, one can generate a 3D embryonic mouse heart in this case. So this provides a very viable opportunity to study cardiomorphogenesis in zebrafish. Once again, the sample can be pressed in this tube, the cartoon to demonstrate this perpendicular pathway. And as I shown before, this process allows us to visualize the dynamic process of cardiomorphogenesis at different stages in 2 dimensions, or this is a 3D. And I think more importantly, if you look at panel G, allow us to track the initiation of a trabeculation or the inner muscular ridges of the heart, starting at 50 hours and up to 70 hours or later, so you can appreciate how mechanical forces in combination with intracardiac flow and contractile forces and working in concert to promote the proliferation and differentiation of cardiomorphic, differentiation of trabecular muscular ridges. All right, but to do this requires some 4D reconstruction of the contracting heart. So we apply the synchronization algorithm and working in concert with Professor Quad at USC to address this and synchrony, because each heartbeat might not be perfectly in synchrony with the next one, and so how we can overlap this irregular cardioperiodicity to generate better contrast. Right. So I think these are the work done, published already, so I'm probably not going to spend time, but essentially this is the very published synchronization algorithm involving period determination followed by shifting determination and then post-processing. Okay, so I'm going to skip this part in the interest of time. Just to show you how this concept can improve the imaging quality. So this is showing the pre-synchronization algorithm in terms of time domain and y-axis. So this is for example, this is the atrium and ventricle, okay, before synchronization algorithm. And then after synchronization algorithm, one can have much improved contrast for spatial and temporal resolution, leading to disk differences before synchronization and followed by synchronization, and that provide the mechanism to generate better intracardial trabecular ridges. And the strength of building light sheet ourselves in the lab is to be able to modify the light sheet fluorescent microscopy tailored to different organisms, such as embryonic hearts versus zebrafish hearts. Well, so basically, this laser light sheet is really based on generally based on the principle of this confocal region, as well as and the thickness expressed in terms of omega naught, and the distance between omega naught and omega is expressed in Z. So this light sheet thickness and light sheet length, and these two parameters can be manipulated by the optical component and in terms of to generate the thickness of these laser sheets, and that impacts on the spatial resolution of the imaging quality. So shown here is the example for embryonic zebrafish. So one can generate the confocal region to resolve to 4 to 5 micron. This is the adult zebrafish, and then we can also modify this light sheet thickness and length to adapt to neonatal mouse hearts. So with this idea, this is the setup we have in different combination. In order to provide more light penetration, instead of having one light source, now we can have two light sources to provide deeper light penetration for a large sample size, such as rat hearts. All right, so this is the optical setup, similar to what I discussed before. So, and then in addition, the idea of subvoxel light, so we can also image the sample from different angle and then reconstruct these images for better light penetration to generate a so-called super resolution. So shown here is before this algorithm, and after the super resolution, one can resolve the neuromuscular junction in zebrafish, or in the mouse rat heart, you can really resolve the ventricle and also the trabecular ridges by this super resolution approach. All right, so that's just more example as to how this process can resolve the endocardium. So if you so this is this laser light sheet cutting across the mouse hearts. And then during this process, one can resolve different structure in the endocardium, and one can also visualize the epicardial coronary arteries, as well as the valvular structure, and in the right ventricular outflow tract and left ventricular outflow tract. And this is the atrium, and one can appreciate the muscular ridges in the appendages. So this is just one example of how this light sheet system is capable of providing very high spatial resolution. And shown here is the aortic arch and calcifications. All right, so now go back to this cartoon illustrating the focus to study initial trabeculation. And the reason for doing and for study trabeculation is really inspired by the clinical manifestation, the so-called left ventricular non-compaction cardiomyopathy. And patients that develop this non-compacted cardiomyopathy will have very thick intramuscular ridges, as shown by this MRI video. And shown here, these are the trabecular ridges. The gross morphology further corroborate these highly trabecular muscular ridges in both right ventricle and left ventricle. Let's notice how interconnected these trabecular ridges are in both chambers, and they are non-compacted, and so that they will result in developmental issue in terms of contractile function and also electrical conduction dysfunction. So in zebrafish, I think the gold standard is what Dr. Vermont is working with transgenic zebrafish line. But then if you would like to quickly screen different genes, morpholino is one approach to silence the protein. So one can use the GATA1A, similar to your +1 paper where you have the GATA1A knockout model, but this morpholino will reduce numbers of red blood cells and thereby reduction in viscosity and shear stress. And so that one can use this approach to study the effect of trabeculation and in the absence of red blood cells, or using NNT2 morpholino to impede cardiac contraction, leading to lack of contractile forces. So both manipulation, either reduction in viscosity, leading to reduction in shear stress, or reduction in contractile forces, leads to the absence of trabeculation. Therefore, therefore we ask the question: what could be the potential mechanistic pathway leading to this phenomenon? Right. So the light sheet corroborate the wild-type trabecular ridges, but in the presence of reduced shear stress, trabeculation is attenuated in this. Whereas, but by the same token, lack of contraction also attenuates trabecular formations. So this diagram illustrates the working hypothesis where the endocardium and myocardium interacts together in the presence of shear forces. These shear forces activate, for example, notch signaling, in this case, ligand receptor interaction, leading to activation of ERBB2 expressed in the cardiomyocyte, followed by differentiation proliferation. And so with this pathway has been studied by multiple investigators, including your group, as well as Neil Chi and Didier's group, that might be implicated in these trabecular ridges formations during cardiomorphogenesis. Right. So the idea of this one is the beauty to demonstrate how we can use morpholino to control contraction and also viscosity, and then these are the diagrams showing different processes, which is well known to the group. For example, you can increase viscosity by doping with erythropoietin to increase viscosity, or in this case, using morpholino to reduce viscosity. So one can see different types of red blood cells present in these circulations. So this model allows us to demonstrate this hypothesis that shear stress modulates trabeculation by way of notch signaling. And obviously, the recent science work by Dr. Vermont further illustrate the connection between piezo-1 calcium influx leading to MFAC. But here, this is the early initial work in 2016 demonstrating the the combination of Leishman imaging and morpholino to manipulate cardiotuberculations. So shown here is how we introduce GATA1A or TNNT2 to attenuate tuberculation. And just more example showing how this 3D architecture zebrafish tuberculation at 75 hours and 100-hour post-fertilizations in response to GATA1A or rescue GATA1A post-MRGG1. And so showing that viscosity is indeed implicated, and when we can also rescue this process. All right. More diagram illustrating this notch activity that mediates this tuberculation. This is the wild type and showing embryonic cardiomorphogenesis starting at 2 hours and up to 5 hours. And then in the presence of endocardial notch activities, one can appreciate this tubercular formation at 3 days, 4 days, and 5 days. But when we perform loss of function, in the case by knocking on notch signaling, you can see the absence of the green fluorescent and also the absence of tubercular formation at 5 days after fertilization. In terms of gain of function, by rescuing this process with a notch intracellular domain, and one can rescue this notch expression leading to tubercular formation. So this is just a humble journey to demonstrate the dynamic changes of hemodynamic forces through notch signaling to promote tubercular formations. In collaboration with Alison Marston, we perform computational fluid dynamics with a fluid structure interface. And shown here is we're interested in understanding we don't have the whole answer yet. I think Dr. Yap will be the one who could help answer this question as why tubercular ridges formation takes place in the presence of oscillatory shear stress and positive flow. How will these two distinct flow characteristics modulate lateral induction of notch signaling or lateral activation and to promote ridge formation? Well, the initial work was to demonstrate this hemodynamic structure by segmentation of the structure for registration and later on as computational fluid dynamics. So we can study shear stress profile in the press and acting on the trabecular groove versus trabecular ridges. So the green curve indicate the increasing shear stress acting on the trabecular ridge in contrast to low shear stress over time in the trabecular ridges. And that allows us to generate this genetic manipulation with GATA1A morpholino or V mutant to inhibit atrial contraction or ERBB2 inhibitor to inhibits cardiomyocyte proliferations. And so now one can study the cardiac cycle in the presence or absence of trabecular muscular ridges in response to reduction in shear stress or in response to the absence of atrial contraction and in response to inhibition of cardiomyocyte. So one can assess the time average shear stresses over this genetic manipulation, providing some initial understanding of biomechanical forces underlying trabecular patterning formation. Well, segmentation is a difficult part for computational fluid dynamics. So we actually developed machine learning for auto segmentation, which is very trivial, which is very tedious, time-consuming. By this new method, the so-called SAC transform-based machine learning, reduce the numbers of training necessary and to maximize tuberculation machine learning for muscular ridges, and so that this leads us to perform how we can coupling hemodynamic flow with myocardial vectors using machine learning. And I will explain this later in my later slides, is how this allows us to identify individual factors of the myocardium in concert with the red blood cell in the intracardium to generate a dynamic pattern for intracardial flow in concert with myocardial contractions. Well, the segmentation also provides us to perform virtual reality so that one can travel inside the zebrafish atrium. Now you are entering the AV valve, so one can measure the diameter orifice, and then one can continue to enter into the ventricle, as you can appreciate the trabecular ridges in the cardiomyocytes. I think in the interest of time, I'm going to finish this video and move on to the final conclusion of our initial study using light sheet imaging. And so you can appreciate this process of trabecular formation by way of notch signaling and to study cardiocontractile function. And I think a combination of light sheet and machine learning further provide us to understand sprouting and organization of trabeculations. And that Neil Chi and many people, including your group, as well as Didier, are focusing on. Okay, so this initial part provide us with some understanding of how pulsatile flow versus oscillatory shear stress coordinate cardiomyocyte spotting and trabecular organization. And given the time we hope to share with you next time, that this trabecular ridges actually play a dual function in addition to provide oxygen diffusion during cardiomorphogenesis and also later on endothelialization for coronary arteries, but also provide kinetic energy dissipation to maximize contractile efficiency. So this is just the concept of how this trabecular ridges could address kinetic energy dissipation between systole and diastole in response to GATA1A morpholino or ERBB2, showing this distinct genetic energy dissipation between the wild type and genetic manipulations. So kinetic energy for contraction versus dissipations. So you can see the lack of dissipation can also impact the cardiomorphogenesis and diastolic function. We hope to share this data in the future with your group. Likewise, genetic manipulation of trabeculation can modulate the strain rates. And just to demonstrate, again, the difference between wild type and presence of GATA1A morpholino or ERBB2 inhibition versus real mutant to inhibit atrial contraction. So one can appreciate distinct differences in terms of volume and strength and during cardiac cycles. All right, and so therefore we use the in silico simulation. We observe that trabeculation promotes kinetic energy dissipation. I think we need more data to further support how this kinetic energy dissipation will be able to preserve ventricular contractile function in the future. And however, the initial data suggests that the absence of kinetic energy dissipation in a non-trabecular ventricle engender ventricular remodeling and loss of ventricular functions. All right, I think with the remaining time, I would like to focus on alpha track. I think we learned a lot from your group. Your group definitely is spearheading this direction, and we are very humble to learn from your amazing work. And so this is the light sheet imaging showing how we can capture the valvular formation in zebrafish. Shown right here is the light sheet imaging of the transcending zebrafish line. M. Cherry labeled the CMLC cardiomyocyte light chain for endocardium, and the FLIC1 GFP labels the endocardial layer, in this case, the ventricle. So using this model, we were able to study cardiomorphogenesis in terms of changes in ventricular volume by using light sheet model and also in concert with 2D computational fluid dynamic, coupled with a fluid structure interface in collaboration with Alison Marsden and her former postdoc, Vijay Vendula. It's a complicated slide, but I just want to show you a couple of examples how genetic manipulation, in this case, this is the control as well, but in terms of pharmacological manipulation, such as metoprolol, beta blocker to reduce contractility, or BDM and to inhibit contractile function, or isoproterenol to alpha-1 and beta-1 agonists to increase contractility and heart rate, or doping by giving erythropoietin to increase viscosity, or by activating Notch signaling by introducing Notch in cellular domain, or GATA1A morpholino, well known to your group. So I think just show you one example where this is the control where one can appreciate the bubble arterial, the valve between ventricle and bulbous arterial versus the VB valves. And notice that VB valve during systole and diastole, and cartoon illustrate the morphology. This is the example of doping by increasing viscosity and leading to increasing length of the valvular formation as well as volume. So we can quantify this in these bar diagrams. And in contrast, if you were to inhibit contraction, notice the absence of valvular formations. So I think we don't have the transgenic model, yet this very quick genetic manipulation or pharmacological intervention allow us to make the observation of mechanical forces, contractile forces, or intracardial flow that might modulate valvular formations. I think we are also, the next question is if you can really follow this valvular formation by developing these, using our UB switch line to study to track the endothelial cell during cardiomorphogenesis. Right here, you can see that this is the endocardial lining in the alpha tract. And then so at 6 days post-fertilization, one can, by this UB switch, one can visualize KDLL, which is the endothelial cell receptor, in cherry. Further, more 8 days after post-fertilization, this endothelial cell are present in the VB valves or in these alpha tract regions. So I think hopefully this model will provide the transgenic model to study alpha tract formations. And shown right here is the confocal imaging, and to illustrate the SNEL1 expressions with CMLC and cherry in terms of the CMLC expression in the alpha tract. So again, this is the atrium, ventricle, alpha tract. So the red arrow indicates where the alpha tract is and where SNEL1 expression taking place. And by noting we have this, when I read your postline paper, I think in 160 hours, I could see that your SNEL1 expression disappear using your RNA scope. But here we noticed that this SNEL1 expression disappeared after 144 hours post-fertilizations. And then in the presence of TNNT2 to reduce contractility, we noticed that this TNNT2, the SNEL1 expression was not observed in our confocal. And likewise, isoproterenol, in contrast, to promote contractility, and also further allow us to visualize these SNEL1 expressions shown right here. All right. So I think we demonstrate using staining methods and to show that this pharmacological contraction or genetic manipulation modulate endo-NT as a potential mechanism underlying valvular formations. So the endocardial shear stress induced Notch1B endo-MT mediated VB valve hyperplasia in this study. And then whereas decrease in contractility results in the attenuation of Notch1B activity, application of endo-MT by our staining approach, and the the lack of formation of a VP valve in the outflow tract. I think we'll still study how SNEL1 could be implicated in this case. So this is just another imaging approach and showing the SNEL1 expression. And I think analogous to your paper here, we observed that SNEL1 EGFP is expressed in the not the endoluminal side, but in the upper sites and during cardiomorphogenesis. And then it's also linked with AP2 genes and using the RNA sequencing approach. And this is just another diagram to demonstrate, I think in the interest of time, I'll bypass this, that Notch signaling is implicated in this valvular formation. And furthermore, I think in the future, the string protein in interaction map could further allow us to connect the mechanical forces and in terms of Notch signaling activation, in this case, the mouse model, delta, you can notice the delta 2, delta 3, delta 4, and jockey 1, jockey 2, Notch 2, Notch 1. And this Notch signaling may be implicated on ERBB2 as well as SNEL1 and SNEL2. And by the same token, in zebrafish, one can also recapitulate Notch signaling and leading to SNEL1B and SNEL2 formations. I think this work has already been done by Dr. Verban in your previous eLife publications. So to summarize, our humble journey suggests that ventricular contractility and wall shear stress work in concert to promote VB valve formations. And so we use our humble approach, morpholino as well as pharmacological approach to demonstrate these initial formations. And I think the next stage is to really understand how this transcriptomics underline this exciting and dynamic problems. Okay, I'm going to skip this one. And I think maybe I can just use 5 minutes to introduce light field, and then I will open for questions. Will that be all right with the audience?
Yes.
Okay, so light field, so we talk about light, so the first two lectures talk about cardiac circulation and outflow tract. And so now we'd like to introduce the 5D integration of light field so one can study both contractile function and intracardial form simultaneously. All right, so the problem with imaging, as you see using this photo, is the depth of to capture and all the faces in this, right? So if you look at a small photo, you want to be able to catch the faces in the front, but also the faces towards the end. So how would you capture dynamic blood flow in this fashion requires a different approach. Therefore, instead of using light sheets, now you can use light field to provide volumetric imaging. So you can capture the whole snapshot in one single frame. So I think without going over the detail because of time, so the idea is now to be able to capture the whole volume of images without cutting 2D slices in the case of light sheet imaging. So I will show you how this is being materialized in our lab, but just some fundamental concept about light sheet where you generate basal beam. That basal beam, as I show, can generate this thickness that determine the resolution of the your sample. Whereas the introduction of light field is to provide volumetric detection. So now you use the basal beam instead of Gaussian beams, and that basal beam allow you to capture this area, this sample in 3D as opposed to 2D. All right, so that allows you to capture everything simultaneously. Whereas in the case of Gaussian beam, you generate a 2D images, then we have to perform a 3D reconstruction and in a time-dependent manner. Whereas in this case, you provide volumetric capturing and all at once. Okay, so this idea of generating multiple light beams allow us to generate volumetric imaging. So I'm just going to quickly compare the differences between light field and light sheets. In terms of spatial resolution, the current light field, current light sheet is 1.5 by 1.5 by 2. However, since we want to capture rapid, volumetric, then the trade-off is a spatial resolution, 4 by 4 by 10. All right, but then the imaging rate is the same, 100 volume per second, or even 200 volume per second, based on how fast your CMOS camera, or how much you want to invest in your CMOS camera. And both are based on fluorescent probes. And then phototoxicity is low in contrast to confocal. And the difference is the light field provides 3D volumetric detection, whereas light sheet, selective plane illumination 2D, right? So therefore, the complementary and synergistic strength of these two optics will enable us to improve our cardiac imaging. All right, so let me show you this combination. I'm going to skip this part, okay, because of time, but just to show you how this combination is possible. So right here, this is the light, this is illumination planes. This is the zebrafish. One can generate a light field. So light field right here, you can see the red blood cell. So you can capture this red blood cell in the volumetric pathway in a time-dependent manner. So that when this is type of images we can capture in the ventricle and atrium. At the same time, we can use light field to capture the ventricular contraction, so that one can generate a dynamic ventricular structure to understand local strength variations in zebrafish. So the end result is to demonstrate the ability to study intracardiac flow dynamics as well as ventricular contractile function in this case. And then so we can actually study the vector field of the individual cardiomyocytes during cardiac contraction. So the end result is shown right here. This is the volumetric imaging by light field. So you can capture a volume of red blood cell in a time-dependent manner to reconstruct the red blood cell formation during cardiac contraction. Simultaneously, one can capture the myocardial contraction by light sheets to capture the individual myocardium. And so now we can perform synchronization algorithm and 3D reconstruction. So by merging these two, one can generate the cardiac function in terms of myocardial contraction and intracardiac flow. Shown right here is this realization during diastole and systole, and so that one can visualize the changing dynamic or structure of trabecular ridges during diastole versus systole. So this diagram illustrates this possibility of combination of light sheet and light field to study these dynamic changes during zebrafish cardiomorphogenesis. All right, so finally, I just want to share with you our humble journey in 2014. Light sheet allowed us to demonstrate 2D plane in time-dependent manner. And 2015, this 3D plus time became possible with synchronization algorithm. And recently, the combination of light sheet and light field allow us to combine both myocardial contraction and intracardiac flow dynamics. Well, so to summarize, we provide this cardiac imaging approach to study myocardial contraction. So we use machine learning, providing post-imaging registration and segmentation for co-localization and with endocardial versus myocardial notch signaling. And we also briefly talk about how we integrate light sheet and light field to elucidate mechanical transduction underlying cardiomorphogenesis. So this is the end of the talk. I hope we'll see the light at the end of the pandemic in a different manners. And thank you so much for the opportunity to share with you. And these are people who've done all the work. Without these people, I think none of this data can be possible. And finally, thank you for your attention. And we think everything is possible by team science, and we hope to bridge with Imperial College of London in the near future. Thank you so much for your attention, and I'm sorry for being and taking too much time.
No, thank you very much for the very inspiring talk. Are there any questions from the audience? I know I have a few, but are there any from the audience? Yeah, Peter, please.
Hi, it's Peter Weinberg. Yeah, I hope you can hear me.
Yes, hi, Peter. Nice talking to you again.
And you, and a very beautiful talk, absolutely stunning images. And I wondered if I could just take you back to the beginning, because I'm not sure I understood about the combination with the clarity technique. Does the light sheet mean that you don't have to use the clarity method, or are you combining the optical clearing in some way? And if so, how does that affect the properties of the tissue?
Okay. Well, Peter, thank you so much for insightful questions. I think just for the audience, likewise, I didn't have time to slow down. So in this case, if you look at the zebrafish model, since zebrafish is transparent, so light penetration allows us to perform light sheet imaging in real time to capture the dynamic process in light sheets in zebrafish. Whereas in large samples, such as mouse model, in this case, the mouse hearts, or in this case, the vasculature, because of the opacity of the tissue, light will no longer be able to penetrate. So this is the case where for large sample, such as mouse tissue, mouse brain, the use of optical clearance is necessary to render the tissue transparent, and so that the index of reflection matches with that of the light sheet imaging system. Does that answer your question, Peter? For example, this rainbow heart, we have to perform optical clearance. Likewise, this is the aorta of the mouse, aorta with calcification, and to visualize this requires optical clearance, so light can penetrate to the deeper tissues.
Oh, okay, thank you. Yeah, that's very clear. Thank you very much.
So I think the common question is, can light sheet be used for imaging mouse sample? The answer is yes, but we have to do optical clearance. And then the drawback, then the question is, can we be used dynamic images? Unfortunately, because of optical clearance, dynamic images are very powerful in the zebrafish model or other transparent specimens.
Okay, thank you very much.
Thank you.
So it seems like with your imaging of the mouse or even the rat hearts, I could see that you can tell where the myocytes are oriented. What is the myofiber orientation? And one of the very interesting things that we have been talking about in the biomechanics community is how that helix angle and the transverse angle of the myocytes, affects the function, and how that is even formed, in the embryonic process, and what caused it to form in the right way. Do you think that maybe some of your images here can be used to, answer those questions?
Yeah, I agree. So the images we use for super resolution will be able to help with your biomechanical expertise to address this torsional contractile forces. But I think I'm not the expert, and I think you and your colleague in solid mechanics will definitely be, we'd definitely be happy to help to provide the images.
Great. Actually, another quick follow-up, right? Actually, on a different topic is in the zebrafish embryo. And one of the things that I've always find hard to figure out in my head is, we talk about how flow is needed for trabeculation, but whenever we have flow, we have some kind of deformation in the tissue. We talk about how the notch one is, and we showed it quite nicely actually, that is a pathway for the flow to affect the trabeculation process. But then again, notch one is also indicated in deformation of the tissue. Is there any the data that you know of that has been able to dissect whether it's a flow or deformation, or to try to figure out which is which?
Yeah.
As in, which is actually causing the trabeculation to form? Is it a flow, or is it actually the tissue deformational stimuli?
Right. So I think I agree with you, right? So the question is, do intracardial flow dynamics and myocardial contraction work in concert to modulate valvular formation or to modulate cardiac trabeculation? Yeah, we feel very, but we don't have the data yet. But I can just cite the work you have done, the recent science paper where your team, used these microbeads to generate endocardial deformation leading to calcium flux, right? And then calcium flux, in your case, activation of MFAC. But then I think other people such as Didier, Stanley Didier, is demonstrating piezo1 and notch signaling to affect the notch receptor. But then if you were to, so this is funny, and the intracardial side, the flows, the shear stress side. But in terms of myocardial contractions, I think if you were to refer to Nature paper published by Neil Chi in 2016, and where his group proposed this something called lateral inhibition, and where in the cardiomyocytes, at certain stages, myocardial contraction can actually, deformation can actually inhibit, will activate the JAK1 signal to inhibit ERBB2 receptor. So his data implicates that this lateral inhibition due to myocardial contraction could be the mechanism for this trabecular ridges and groove. But I think we need more data, and definitely it's not, so I think that's, I'm not quite sure if I answered your question completely, but it's a very exciting area, and we don't know the answer. But I think working with you and other people, hopefully we'll be able to decipher the role of intracardial flow versus myocardial contractions.
Thanks for that. Are there other questions? Maybe one last one. Sorry that I took up some time. Okay, I guess, yeah, it is about time. And once again, we just like to thank Professor Hsiai so much, for taking time to come here and share with us such exciting and beautiful images. Yeah, it would be quite exciting to keep talking and see how we can work together. And thank you to the audience for your time here. Yeah, we'll see you then.
Yeah, thank you so much again. Looking forward to visiting your institution again. And thanks again for Peter Weinberg for organizing that beautiful symposium 2 years ago. So once again, thank you very much for the invitation.